Thank you, first of all, for having me today. It's a pleasure and honor to get to be part of your seminar, our series, and thank you to Ellen for coordinating and Steve for inviting me to be part of this. And so I'm going to talk about some of the work that I've done, that some of my students have done, postdocs, really with the focus on understanding how climate change is affecting amphibian populations and amphibians at the range level. And before I get going, I want to acknowledge the people and the funding and agencies that have been part of this. There's been support from a wide range of entities, and below on the bottom, these are my grad students and postdocs in the last couple of years who have all been part of understanding and our research on how climate and land use change is affecting wildlife in general. On the top three are three of my grad students, all PhD students who now finished their PhD that have been specifically involved in this research. So on the left, Dr. Stacy Ambergy, who's now a postdoc at the University of Washington. In the middle, Dr. Courtney Davis, who's a postdoc at Cornell University. And then on the right, Dr. David Munoz, who is doing some postdoc work with me right now. Beyond my group, the results I'm going to talk about today have come about through collaborations that have involved dozens and dozens of different people. And in fact, Stephen is one of those for one of the topics I'm going to talk about today. And in their research on the continental level, trying to understand how amphibian communities respond to climate change. So I'm going to talk a little bit too about how other approaches we're using relate to harnessing and applying collaborative networks to answer questions. So I'm going to talk about amphibians, but in general, as I said, my lab and research group, we're focused on wildlife, terrestrial vertebrate species, and how they respond to climate change, and really focusing on the population and community level, and working across a bunch of species. But one of the questions we've been focused on and interested in is, all right, so maybe an issue with the mic. So I'll try and make sure, talk a little bit louder, and hopefully that'll clear up. Let me know if still having issues hearing me. But one of the questions we've been interested in is how do we predict range shifts? So this is one of the major responses we expect for species and climate change, is that we're going to see shifts in their ranges. And our standard toolkit really harkens back all the way to approaches that were first applied by Grinnell and others more than a century ago. And this idea of trying to understand the way that environment structures a range. This is often done through niche envelope modeling approaches, where we look at how the range relates to different environmental parameters. And the idea when we want to predict range shifts is we predict how those environmental relationships or climate is going to change through time, and use that as a mechanism to predict the future range. So that's on one end. On the other end, we have biophysical models that really dig into physiology and try and understand how species respond and how they will respond by basic physiological processes. And these have both been really valuable in trying to understand and make predictions about climate change and how species will respond. What my group has been interested in, and what I've spent a lot of time focused on, it is somewhere in between, which is trying to use a more demographic approach to understand how species are going to respond to climate change. So by demographic, I mean measuring the actual demographic processes at the population level that lead to increases or decreases in local populations, and essentially extrapolating that up to the range level. And this is not a new idea. This has been suggested by others, such as Janet Franklin here more than a decade ago, recently in Mark Urban's review on forecasting and climate change from a couple of years ago, I believe in Nature, have all suggested this as an approach. So if we want a demographic approach, we can think about what leads to range shifts. And when I think about this, I think about the fact that shifts in range are really an outcome of changes in local populations. So the reason we get a shift in range is because some populations increase and others decrease. New populations are formed through colonization, some go extinct, and this is what leads to changes. Now, these shifts in these changes in the local populations occur because of local population processes. And we can describe these processes using different approaches, whether it be just the occurrence of a population and metapopulation models, patch occupancy models, or approaches that try and describe population processes, basically based on the occurrence or the presence or absence of a population. Or we can do it with the number of animals in these local populations using demographic sort of processes to describe how these populations respond to climate change. The other key part about trying to understand range shifts is understanding that environment doesn't actually act on populations at climate scales. It's not decadal averages that lead to changes in populations. It's really the short time scale weather events that lead to losses or gains in populations. Now as we get climate change, we're going to get a shift in the distribution of these short time scale weather events. But if we want to understand the process by which a population responds, it's really on a short time scale where environment acts on the actual population. So taking these three concepts, I'm hoping I can demonstrate that we can learn about the potential for range shifts to happen by, one, focusing on local populations, two, describing those population processes, and three, by linking those population processes to real-time weather events. And if we can make if we can understand those relationships, then we can understand why and where and when range shifts are likely to happen. So there is a challenge to this approach, and unlike climate scientists who are trying to understand the dynamics of climate and weather and make predictions on that end, or many other disciplines where we're working on the global level, when we talk about wildlife, we don't have the sensor system that we have for some of these other processes. So we can't take a satellite and measure the number of animals in a population, at least yet. We don't have the sort of global weather observation networks for wildlife that are automated and lead to large data streams. And we're getting better with sensor technology. We have acoustic recorders, we have camera traps now, but even these these methods rely on really intensive post-processing. So we need to be creative if we want to get the data streams to be able to ask questions on the scale of species range. And if we want to ask large scale questions about whole communities of wildlife. So this is a first challenge and one that I think we've been thinking about approaches for dealing with. A second big challenge we have when we deal with wildlife and trying to link predictions and understand how populations respond is the uncertainty in the data we get. And here's three of my favorite birders, my family. They're pretty good, but even when we go out with these three, we don't see everything that's out there. And so the sort of data we're collecting on a weekend birding trip has uncertainty in it. We're going to miss species. We're going to have uncertainty in the observations we need to deal with, and we need robust methods to deal with it. I'm going to talk about amphibians today, and for many of the species we're dealing with, they disappear for most of the year. This redback salamander on the right here, it's a species that's going to spend the great majority of the year underground. These wood frogs here, and I'll talk about them specifically, they're really only observable for a short period within the year, and then they're really hard to find and quantify and try and understand their population. So we have uncertainty and we need to quantify it, account for it, and really deal with it if we want to make predictions. So that's another theme of what I'm going to talk about today. So I have some ideas of how we meet this challenge, and the challenge being trying to develop demographic approaches to understand range shifts. And like I said, we need to monitor local populations, and we need to replicate this across the range. We need to understand the processes, and we need to link it to environmental variables. And to do that, and to meet the data and observational challenges, we really need to harness the resources that are out there. And one of the most promising is community and citizen science. And we're getting there for amphibians. I'm not going to talk about that so much today when I'm talking about amphibian responses, but we do a lot of work on birds too, and community and citizen science has been crucial for the sort of questions we're looking at and answering, and also some work I do with folks who work with insects. The same thing, community science has really been the way we're starting to get the data sources we need. More relevant to what I'm going to talk about today and the approach we've used for amphibians is the development of collaborative research networks. So trying to answer large-scale questions using large-scale collaborative efforts. And I'll talk about a couple of those efforts, actually 3 different ones today. But the other thing we need to meet this challenge is robust analytical methods. We need to link our data collection and inference in ways that allow us to combine data streams and really answer questions in a robust manner. We don't have the sort of robust sensor data we might have if I was a stream ecologist looking at stream flow or other disciplines where I envy their data streams. So we need to be smart about how we use data. So I'm going to talk about 3 case studies. All relate to amphibians and climate. The first one we're going to talk about wood frogs, and a goal here in this research was to understand how responses of a single species differ across its range. So I'll focus on that and measuring those sort of responses. Then I'm going to talk about a second analysis that was more focused on species richness and how communities respond as a whole to changing climate. So less of the within species and more trying to understand among species variation in responses. And then finally, I'm going to talk about some real nascent efforts that we made to build a network from the ground up to understand how a single species, but hopefully a model for other species, is going to respond to changing climate. And I'll talk about that last. All right, so first off, I'm going to talk about wood frogs and measuring within range variation in climate sensitivity. So this is a question we're trying to understand how the species responds across its range. This was really led by Dr. Stacy Ambergy, one of my former PhD students I already mentioned, and work with a bunch of collaborators trying to understand for the single species how they respond to climate change. So the data we used was data on egg masses. So if you're familiar with wood frogs, they come out early in the spring as soon as things thaw. That last slide there, that's the range of the wood frog there in that picture. And they come out in the spring as soon as things thaw and lay eggs. And really, they're only the adults that come to a pool to lay eggs are only going to be there for a few days. But they leave these big, obvious egg masses once they're done breeding. And the data we're using is counts of egg masses. And it happens to be that a lot of different people are counting egg masses, and they've been doing that repeatedly at ponds for a long time. So we were able to build a network of 26 different study areas throughout the wood frog range, where people were collecting egg mass count data. Many of these data sets span more than 20 years. And it allowed us, through this collaboration and working together, to understand, using this metric, egg masses, how these populations respond to climatic variability. So I really quick want to talk about how we sort of formulate this demographic model, and we estimate these relationships. And essentially what we do in this did in the system, and we're going to do again in the second one I talk about, is developed a state space model. So a model that tries to disentangle a state process, how the population size changes through time, and then relate that to what we call an observation process, which is how many actual egg masses do we observe during the survey. And underlying that is a population model, and a really simple population model here. We have an exponential model where the population size in year t plus 1 is equal to the population size in the previous year, times e to r, where r is the instantaneous growth rate of that population. So little r, a measure of population growth rate, we all learn about in basic ecology. We then allow for essentially sampling error. The fact that we can get noise in our observations that has nothing to do with the changes in population size. And we do that through a simple observation, normal distribution error sort of model that works on the actual observations we observe. And this whole approach goes way back to the 60s and methods that were first developed for engineering and understanding the dynamics of systems by engineers using what's called the Kalman filter. And it's been adapted and reformalized for the sort of work we're doing. And one of those sources is Carey and Schaub here from 2012. All right, so that's the basic model. We have a real simple population model to describe changes. And what we really want to understand is this little r and what influences that population growth rate. And so we can actually build a model to begin to understand that relationship. And that's what we did with these data sets. And we tried to formalize some predictions about how we thought the environment would influence the growth rate, and also how that relationship should change depending where we are in the species range. And we put that together, and we have an approach where we can look at the demographic parameter, the population growth rate, and how it's influenced by climate. So what are those predictions we tried to test? Well, here I have the wood frogs range, and this blue dot in the top represents population in the far north part of the range where things are cold relative to other parts of the range. And this green dot is in the south, in the more warm part of the range. So if we think about that and how we think climate or the environment or temperature might influence those populations, what we probably expect in this very northern part of the range is that the warmer it is in any given year, the better this population is going to do. They're already at the cold end of their extreme. So if it's colder than normal, we expect the population growth rate to decline. If it's hotter than normal, we expect that population growth rate to be higher. Similarly, in the south, we're at the warmest part of the range, and what we might expect is that in the warm summers, or the warmest years, it's going to have a negative effect on this population. We're already at the warm end of the range, so more extreme warm temperatures, we might expect negative effects. And these are the sort of relationships that underpin our other approaches for predicting range shifts. They predict that basically if the range represents an ecological niche, then these sort of variation and these sort of relationships should emerge at the population level, but they're rarely tested. And of course, we can look at the middle of the range, and this might be where the environment year-to-year fluctuations don't have a major effect. All right, so we have this data for wood frogs, and what did we find when we tried to test these predictions? Well, I want to show real quick the range or the distribution of where our sites were and where they occur in climate space. And so this figure here on the right shows annual temperature, average annual temperature on the y-axis, and average annual precipitation on the x-axis. And the blue area is the area where wood frogs occur. The darker values on this are basically all of continental United States and Canada. So we can compare where wood frogs are compared to the range of possible places where they can occur, and we can see our locations do occur across the wood frog range, but primarily in the warmer and the wetter part of the range. So we do have observations that do butt up against the edge of climate space, but unfortunately don't go quite all the way across the range. So we're gonna be thinking more about one edge of climate space. And so we took the data from the 26 populations. I believe there are over 700 wetlands in this data set. Most of these data sets span more than a decade, and begin to test relationships and predictions like the one I showed. And sometimes we found results that fit our predictions. And I have two here, and I'm going to walk through one of them. that are consistent with the predictions. And on the left here, we have a measurement, we call it sensitivity, which is simply how does a local population respond to changes in that climatic variability. And on the y-axis, we have the average climate. So if we look on this one on the left, what it says is we get positive responses to increasing maximum summer temperature when the climate is cooler. We get negative responses in areas where it's warmer. And we get the opposite for winter temperatures. So let's walk through that in a little more detail. So if we look at this figure here, where the arrow is pointing, this is the space we're talking about in the more northern part of the range. And in this part of the range, we get a positive relationship between what is the summer temperature, basically the anomaly, is it above average or below average, and the population growth rate. So we get warmer is good in the colder part of the range. Similarly, in the warm part of the range, warm summer temperatures have a negative effect. So we get the relationship we expect there in the middle, and then somewhere in between, there's no relationship. So this fits what we typically think about the response being when climate warms, which is we're going to see range shifts where we get negative effects on the warmer part of the range, and we get positive effects on the cooler part of the range. So we see this as it relates to maximum summer temperature and as it relates to winter temperature. We can actually observe that. But in other cases, our results don't follow the predictions, and that's the case for precipitation. So what we actually found was that even in the wettest part of the range, there was a positive effect of more water. And in fact, there's more benefit of more water in areas that are wetter to start. And this doesn't follow our niche envelope predictions and suggests that things might be a little more complicated when we talk about precipitation. And I think it's actually something we found a lot with amphibians is precipitation, because it relates to hydrology and hydrology in wetland environments is affected by strong interactions with the environment and the sort of the structure of that ecosystem, is we don't get the sort of clean results that we often get for temperature and making predictions about how amphibians will respond. So in this case, for precipitation, it didn't follow our predictions. And in other cases, we found that to really understand how amphibians are going to respond, we need to understand the interaction between temperature and processes related to temperature and those related to precipitation. So for instance, in this case, we find that the benefit of greater precipitation during breeding is greater in areas that are cooler. And again, this isn't what we might expect based on just a priori reasoning. So it again suggests that the way that precipitation is influencing these populations is a little more complicated than temperature. All right, so basic conclusions of that work that we made is that, first off, at least for the wood frogs, I think we're seeing it in some of our other results, is temperature predictions are a lot more straightforward to make about how amphibians respond. In this case, in the warm part of the range, higher temperatures are having negative effects on wood frogs. Those are easier and more straightforward and closer to what we might expect than what we're finding for precipitation. And another result we found in a lot of the work we did with wood frogs is that interaction between temperature and water availability is really important and probably complex. And I think that has to do with wetland hydrology and maybe some of the community interactions. All right, so that's a single species. We try to expand this approach to multiple species, and that's what I'm going to talk about next. And this was work that was funded by the USGS Powell Center. We had a working group of around 15 people, and we set out with this goal of trying to use a similar approach to the wood frogs to ask this question about how are amphibian communities as a whole going to respond to climate change. And the first thing we did is build a network of existing research and data that was out there from many different sources, and collaborated and brought this together into a single database. So in the end, we actually had data from more than 80 study areas, more than 80 species, more than 600,000 total observations collected by over 25 research groups that contributed data to this effort. And the commonality in the data we use in this analysis is like the wood frogs. It's observations of populations that are collected repeatedly over many years. And the way those observations were collected differed. It included DipNet surveys for amphibians, visual surveys, acoustic surveys, coverboard surveys for terrestrial amphibians. But what was common between all the data I'm going to talk about here in a second is this common framework where in every case we have a study area where data collection happens. So here we have 3 different study areas. Within the study areas, we had multiple replicated locations where data were taken, whether they be wetlands or stream reaches or terrestrial plots. Multiple species were observed across multiple years. And for each species, we get multiple observations within a year. And this framework allowed us to one, generate the sort of variability among species and among sites to ask really interesting questions. It gave us the temporal time series to ask about dynamics and how populations respond. And it gave us the replicated data we need to actually address questions of observational uncertainty. So what we did is we took this data and we used it to estimate, in this case, a simple patch occupancy model. So wood frogs, we have a population model, essentially an exponential model where we look at abundance through time. Here we're looking at the presence, absence of the species at a site. And you can imagine if we're doing this for many species at the same time, we begin to be able to ask questions about not only the distribution of individual species, but also species richness at individual sites. So again, we think about climate anomalies, so year-to-year variation. And here, just for illustration, I have a 5-year time series of the soil moisture anomaly. So negative values being drier conditions, positive values being wetter than average for a site. And our goal was to link these anomalies to changes in year-to-year status of whether a species was present at a site. So we can imagine in any given year, a species may either be present or absent, and that status changes from one year to the next. And we want to know how these anomalies, this year-to-year changes, influence the changes in whether the species is present. So that's the basic structure of the model, with one more component I have here on the bottom, which is statistically accounting for incomplete observations. And the way we do that is through the replicated samples. So in any given year, we might get multiple observations of whether a species is detected or not. So in this first year, the species is not seen, it's seen, then it's not seen. The second and third year, it's never seen, and then maybe in the fourth year, it's seen during all three observations. And our uncertainty lies in this year and two and three, because that species still may be present, but we might not observe it. And the way we deal with that is essentially estimating the detection rate. We can get it through observations like this first year, where we know, all right, in this case, we only observed it 1 out of the 3 times we went out there. So we know there's incomplete detection. All right, so the final component to this and how we structured this and asked questions is we were working with, as I said, more than 80 study areas, more than 80 species, and we needed a common set of climatic variables to begin to try and understand responses from. We chose 5 based on a of course, oversimplified, but a generic amphibian life cycle, and key parts in that life cycle where we thought climate would be important. And that includes during breeding, how much water is available. During the peak of summer, how severe or extreme the warm temperatures get. So what is the maximum 10-day average temperature? And also how much does the soil dry out, which is an index both for terrestrial amphibians, but also relates to streams and wetlands and the hydrological measures there. During the winter, how cold does it get? So we use what's called an air freezing index. And the amount of snowfall. So we have 5 variables that we're able to use large-scale climatic data sets to ask questions about how year-to-year fluctuations in these variables influence the species in our data set. So we picked this data apart and approached with different species traits, site traits, and asked how these influence these 5 climatic factors. I'll just show you a couple just to sort of understand the backbone of this model I'm going to talk about. So here we have on the bottom the 5 factors: winter severity, maximum temperature in the summer, soil moisture, snow in the winter, and water availability during breeding. Our data, we can divide it to 4 general regions. So in the east, the northeast versus the southeast and eastern forested systems. We had a fair bit of data from western montane systems, which is shown in purple. Then most of the rest of the data either came from desert, grassland, or other dry ecoregions, so we lumped it there. We have a few study areas that fell outside of this, so don't show up in this figure. But we'll just look at one of these, water availability. And interestingly, we see, maybe not unexpectedly, in western dry systems, water availability has an outsized positive effect on amphibian populations. So strongest relationship we see. In the southeast, we actually saw a negative relationship here, which at first was a little bit perplexing, but I think it actually makes a lot of sense because most of our data in the southeast came from coastal systems, Florida, Alabama, Georgia, not complete, but a lot of them. And what we saw during the period when many observations occur is some extreme hurricane and other weather events. So we get a negative effect on basically precipitation on breeding, during breeding in this region, and I think it's the exposure to extreme events. And we did the same for other variables. So in the Northeast, soil moisture has large benefits. More severe winters, colder winters seems to benefit, especially montane systems in the West. We looked at other things such as species traits, so variation among species. And here we've separated out the frogs and toads versus the salamanders. But what we see in both groups is SBL, which is a measure of the size of an individual. The bigger the amphibian, for both groups, so the center plot here, the more positive effect we get of greater soil moisture. So it appears larger amphibians seem to be more sensitive to things getting dry during the summer. So we did this, and there's an underlying a bunch of other factors that lead into predictions, but what we get in the end is the ability to make inferences about how amphibian species richness in geographic space, climatic space, and across phylogenetic space is going to respond to changes in these climatic variables. So one of the most universal responses we found was that colder winters benefit amphibians across all the regions. So this figure here, we're seeing positive values as winter severity increases. And I think if you flip this to the analog of this, it's easier to conceptually understand, which is warm winters have a negative effect on amphibians. Cold winter is a benefit, warm and negative. So warming during the winter seems to be a particular issue across the different regions in our analyses. One of the predictions that maybe is most consistent to what we'd expect, so maybe it's not super interesting, but is very strong in what we saw is how the amount of water during breeding period influences populations. And here's another figure I have in climate space. So on the y-axis, I'm showing the mean annual temperature. So higher values are warmer climates, lower are colder climates, and the x-axis is average annual precipitation. And so blue is a positive effect of breeding water availability, a strong positive effect. Green is a weaker positive effect. And yellows and oranges and reds are negative effects. And what we see is that in the warmest and driest regions, we see the greatest positive effects of essentially more water during the breeding period. Interestingly, we see the negative effects also where it's warm, but where precipitation is high. And that again is the southeast. So this is this thing we were picking up in the southeast where at least during the period we looked, greater water during breeding was having negative effects on populations. And then for the rest of the area we worked in, we're seeing still benefits for more water during breeding, but not nearly as strong as in these hot and dry areas. We do the same across phylogenetic space. I don't think the patterns are strong here, but we're able to look at whether different groups were responding differently and see if we could make conclusions based that way. And in the end, I don't think we have a lot of strong inferences that way, that different amphibian groups are responding differently to climate. But what we are able to also do with all this is look retrospectively and ask, based on the climatic change we've seen in the period of 1982 to 2012, what did we expect to be the responses of the populations we were looking at? And this has allowed us to ask, based on what we're seeing, do we expect climate is already having negative effects on populations? And here the red areas are where we think the changes we've already seen are having negative impacts. And that's especially the case in Western montane areas and some other areas in the West. And we see scattered other spots where we might be seeing negative effects, but a lot of the country, we actually expect the sort of changes we've already seen might be benefiting different amphibian populations. And we're able to actually use this information and correlate it to observe trends in these populations. And we see these predictions correlate with what we're actually seeing. So we're, at least in the data we've looked at, there are stronger amphibian declines in Western mountain regions and other places where this sort of model is predicting recent climate change is having negative effects. And then what we found on average is there's a little bit of a negative effect of the climate change we've seen already, but on average, about as many regions or populations were predicted to have positive changes as negative. So this doesn't explain another thing we're seeing, and I didn't talk about, which is across all these different data sets, essentially large rates of average decline. And that doesn't seem to be climate driven. So what's the takeaway? Well, the variability among regions might be related to climate, but climate is not, at least at this point, explaining why we in general see large amphibian population declines. All right, so species richness is sensitive to climate, whether it be positive or negative, whether it be breeding water availability or winter temperature. And we see some really strong patterns, but they don't explain overall climate or amphibian declines. All right, I'm just going to spend a couple minutes here and talk about a new system we've been working on. It's a collaborative network of sites throughout the Northeast. It was an effort to ask these same questions, but to be able to design a monitoring network from the ground up. And it started in 2014 with 4 study areas in Pennsylvania, Maryland, and Massachusetts, and it's expanded to over 25 study areas in the Northeast. And our focus has been on the species here in the bottom left, the redback salamander. And the idea here is we've set up study areas with replicated plots. So we block them, we replicate them within a study area. So this is one of our study areas in Pennsylvania. And on these study areas, we've set up coverboard arrays where we monitor populations of redback salamanders. And if you know anything about redback salamanders, they're a terrestrial salamander, and they'll readily use cover objects during optimal times of the year as refuge during the day. So we can go through and turn over these boards here. There's 50 in a plot. We can catch the animals that are under them, and we mark those animals and we monitor them through time. We're doing this in more than, I think, 25 areas right now. Since 2014, we've captured over 35,000 salamanders across all these different study areas, and started to follow them through time, allowing us to ask questions about their demography and how they respond to the same things I've been talking about, for the last 40 minutes. And And we're beginning to be able to ask really specific questions from having this network of data. And one of them has been about growth and body size responses in amphibians. And there's some data out there that suggests that amphibians and maybe other species, one of the responses we get to climate change is decreases in body size. And so there's this paper by Nicholas Caruso from 2015 that tries to use historic data to understand whether that's the case. And so one of the things we've recently addressed with this data is asking the question, using time series data for individuals, are they responding to climate in ways that's affecting body size? So what we do from the data we collect in these plots is we fit a real simple growth model, a von Bernal Amphi growth model. So this is classic for fisheries. It also gets used a lot for amphibians and reptiles as a way to look at how the size of an animal changes through time. And the idea being an animal will grow fairly rapidly at first and eventually asymptote out at a body size. And so we're able to use mark recapture data to ask those questions. And we fit this equation or model to this data to estimate things like L infinity, the asymptotic body size of an individual, the how much growth is left to get to that body size, and then importantly, how quickly do they grow? And this K parameter is a measure of the slope or the rate of change. So it gives us information about how big animals get, but also how quickly they grow. And these are important demographic parameters. They relate to the health of this population and how they respond to climate. And we're interested, does temperature influence these relationships? And really quickly, we do see patterns, but they're not necessarily what we expect based on other predictions about body size shrinking. So what we do see is actually in the warmest part of the range, females, their asymptotic body size is larger than in cooler areas. And this is reverse to that prediction I told you about earlier. But we also see that the growth rate, or how quickly the salamander grows, depends on also that location. So in the warmest part of the range, both males and females, but especially females, grow more slowly. So they get bigger, but they're slower to get there. And so what we're seeing perhaps in warm parts of the range is this constraint of growth. They might be seeing optimal growth at sort of middle range sort of temperatures. I'm running short on time, but we've been able to ask other questions, such as the metabolic costs of longer summers. The take home here is actually in the southern part of the range. The salamanders seem to have some metabolic plasticity. They're able to shut down in ways that have cost savings during the hottest and driest part of the year. Unfortunately, if we extend into the future, what we think future climates look like, we don't think, based on what we're seeing already, that they have adaptive potential or that sort of plasticity to manage even more extreme environments. So they might be extending their range now, but unlikely to be able to harness that plasticity in future climates. So essentially this 30% is our cost savings by current plasticity that we're seeing already. But this triangle on the right is showing that as we go into future climates, we're not seeing additional cost savings based on plasticity. All right, and real quick, I think what's really fun and interesting and what we've been able to do by setting up this network is we've actually been able to also link research questions with education and outreach. So this network is 25 study areas, is primarily run by faculty at primarily undergrad institutions or nature centers and environmental centers. We've had dozens of undergraduates that have been able to develop research experience at these sites and actually been able to develop classroom tools that allow us to use the research that's going on in these plots as a mechanism to give undergrads in a classroom setting research experience, both in going out to the plots, but actually analyzing the data and making hypotheses and being able to test them. And that includes a bunch of modules we've developed that are being used in different classes by different investigators in this network. All right, so that is what I had today. And I thank you again for having me. I'll leave you with my, after talking about education and that part, my favorite assistant and learner, my daughter Clementine. And if we have time for questions, I can take them.